SwayamPrabha. Digital India. Educated India.
Hello and welcome to our 10th lecture of our NPTEL course Introduction to Mechanobiology. So, in last two lectures I have been discussing about how we can probe protein unfolding using AFM and how we can use that for studying how forces affect the unfolding dynamics of proteins, okay, and taking the specific example of fibrinectin. Okay. So, let me first start by doing a brief recap of what we observed or what we discussed in last class. Okay. So, one, I introduced sawtooth profile. Okay. So, when you do protein unfolding studies with sparse concentrations, okay, and you pull a protein with an AFM, okay, so your sawtooth profile looks something like this. Okay. So, in this case, so what you see is there are 4 peaks. Okay. So, what happens? How do we assign these peaks? So, I said that this first peak is associated with detachment of protein from surface. From the surface. Okay. And the last peak corresponds to detachment of protein from the tip. Okay. So, essentially the middle peaks, these 2 peaks are your data peaks for your protein of interest. Okay. So, typically let us say you get a total of 10 peaks in a given experiment. Then you can bring it down, you can break it down into 8 plus 2, because these 2 peaks corresponding to the first peak and the last peak are not protein unfolding events, but these are detachment events. But these 8 correspond to protein unfolding events. Okay. But it is not necessary. So, for example, let us say you did this pulling experiment and you got 8 peaks, but it is not necessary that you will always get 8 peaks. Okay. And the reason for that I explained in last class was when your protein is adsorbed, you have no control where your tip hits the protein. Okay. So, once the tip starts going up, you have an overhanging portion, this does not unfold. Okay. Only this segment which is being stretched, this is what this unfolds. Okay. So, you can have, so for a protein with M folded domains, okay, you can have less or equal to M plus 2 peaks. So, at max you will have M plus 2 peaks, where the tip actually contacts the one end of the protein, but in all other cases you will have less than M plus 2 peaks. Okay. So, this is one thing to be noted. Now, let us say, so fine, you got your peaks, right. First thing is to record the data, but now it comes to analyzing the data. Let us say you have a protein with M domains, okay, and you get force peaks which have up to m plus 2. So, you know that there are m peaks. So, now the tricky part comes is how do you assign the peaks to specific domains. Okay. In other words, you had these domains, okay, 1, 2 up to m, and you have peaks f1, f2 dot dot fm. So, you do not know whether 1 corresponds to, let us say, f2 or 1 corresponds to fm. So, you do not know that. Okay. So, that is the challenge. How can you assign the peaks to specific domains? Let us take a simplest case. Okay. You have a 3 domain protein, okay, with each domain equally stable. So, equally stable would mean that the unfolding phase is equal, okay, but lengths of the domains varying. Okay. So, in other words, what I am saying is I am drawing these 3 domains. Okay. So, the force required to unfold each of these domains remains the same, but the sizes of these domains are varying. Okay. Lengths of domains varying means the domain sizes are varying. Okay. So, now let us see what the force curve, the sawtooth pattern would look something like. Okay. Okay. So, once again I can dismiss this and these last two, the first and the last peak I can dismiss, but I have 3 additional peaks. Okay. So, 3 peaks. So, because what you see is the magnitude of these forces are the same, okay, but these lengths are different. Okay. So, these lengths are different. So, what does this? So, for a single domain, let us say you have a peak like this. So, there are 2 things that you can determine from this peak. One is force of unfolding, and this is the unfolded length. Okay. So, your x axis gives us unfolded length and the y axis gives you unfolding force. Okay. So, in this particular case, because we took a case where each domain was equally stable. So, your unfolding forces are similar in magnitude, but the unfolding lengths are not. Okay. So, this is one such representative data point. You have to do it multiple number of times and need to determine the statistics. Okay. So, what you plot, okay, Okay. So, what you will have in this particular case that we drew, okay. So, what I have drawn in the, so I have drawn 2 histograms, okay. One is the unfolding force and one is the unfolding length. So, for the unfolding force, because we assume that all the forces were of equal magnitude, that the domains are equally stable, I will get a single force peak. Okay. So, it might have a little bit more scatter because you are collecting the statistics from so many events, and there might be small variations in the standard deviation. Okay. But if these domains are of distinct lengths, you will see that you will have 3 histograms which do not overlap with each other, or the overlap might be there, but the peaks, okay, peaks do not overlap. So, you know, so you have to take this value, okay, these mean values of each of the histograms, and you have to assign this. So, each of these lengths, so let us say L1, L2, L3. So, L1 is the unfolded length okay, of smallest domain. Okay. L2 is similarly the unfolded length of intermediate domain, and L3 is unfolded length of longest, largest domain. Okay. So, what is the unfolded length? If you know the number of residues in a given domain, so then your unfolded length should be equal to the number of residues into, so it should give you the number of residues into 0.38 nanometer per residue. Okay. So, this you can directly find out the unfolded length. Now, because you know the number of residues, okay, because you know the number of residues, so you can calculate this. So, whichever unfolded length that you experimentally measure tallies or is closest to this particular value, then you know you can assign that this particular L corresponds to this domain, okay, so on and so forth. Okay. So, this is how you do the assignment for the simplest cases in, let us say you have engineered polypeptide of the same peptide or things like that, where you have the same repeating unit. Okay. However, this approach will not work if you have multiple domains. So, this approach will not work if multiple domains have comparable lengths. Okay. So, this simplest approach will even work for the other case where your forces, so if forces are different and lengths are same. So, if you knew a priori, so a priori information about domain stabilities may be used for assignment, assigning peaks. Okay. But clearly for this case, this approach will not work if you have multiple domains which have comparable lengths. Okay. So, what is done in these cases is actually using protein engineering, where you make a polyprotein. So, you have a protein with the same unit, same domain repeating. Okay. Now, let us take the case of fibronectin. Okay. So, the FN3 itself has 15 domains. Okay. Now, if you pull this long a protein, okay, and you have C position. Okay. If you pull this long a protein, one thing that you observe, okay, one thing that you observe is you will get these multiple peaks . Okay, so on and so forth. Okay. I am not drawing all 15 domains, but you will have these multiple peaks which might have comparable lengths, comparable forces, so on and so forth. So, it is near impossible to assign the peaks. So, for this reason, what you have to do is you have to construct proteins, you have to engineer proteins which have, let us say, domain 1, okay, FN3, this domain is repeated, and so on and so forth. Okay. So, you prepare this, you engineer these proteins. Okay. So, when you engineer these proteins, so you have to engineer proteins which contain one or multiple domains repeating. Okay. So, let us take one such case. Now, let us say you have made your protein. So, now one important step is, after you have made this polyprotein, you have to make sure you use circular dichroism and other techniques to make sure that the protein folds and is stable. Okay. So, this you must check before you do any of AFM. You must check that the protein that you engineered folds stably. Okay. After you have done that check, so let us say you have made a protein which has this unit repeating A, so on and so forth. Okay. So, typically if you choose a longer length, there is a greater likelihood that the protein will fold, okay, versus making just 2 A's. But let us say you did that AFM experiment. Okay. You did the AFM experiment, okay, and you saw no signal at all. So, you have this situation, no signal was observed. So, in this particular case, what might be the possibilities? Okay. One possibility is, let me draw. So, let us say that the protein is very unstable. Okay. So, to begin with, it does not fold properly, and that is why I said that we must make sure that the protein folds and is stable. Okay. But if you have done this check using biochemistry tools, okay, then you know that this is not the case. Okay. But this might be one possibility, that the protein is very unstable. What might be another possibility? The protein is super stable and requires high forces to unfold. This is somewhat counterintuitive. Why am I saying? So, if we know that the protein is stable, why are we not able to detect it? The reason is as follows. Okay. So, you have your substrate and your protein is anchored, right. So, when you pull up the protein, okay, so you reach this configuration, okay, you reach this configuration, let us say. Okay. So, the tip is moving up and you are trying to pull the protein. So, there is the tip will be able to pull the protein only if the force required to unfold a domain. So, this will work. Okay, let me write it down here. Okay. So, you have this situation. So, protein can be unfolded only if the force required to detach protein from tip is greater than the forces required to unfold individual domains. Okay. Okay. So, this is the criteria. So, let us read it again. Protein can be unfolded only if, so when I say protein can be unfolded, I mean that the unfolding signature is observed in the experiment. Okay. So, protein can be unfolded only if the force required to detach protein from the tip, right. So, this is a system of springs connected in series. So, it is always that the weakest link breaks fast. Okay. So, because the weakest link breaks first. So, if the force required to unfold a domain is much greater than the force of adhesion between the tip and the protein, then the tip will just detach from the protein. Okay. So, you will not see any signal. Okay. You will only see two detachment peaks. So, in this particular case, that is it. This is all that you will observe. Okay. So, this is associated with detachment of protein from the surface and this is associated with detachment of tip from the protein. Okay. So, in this case, so if your protein domains are very stable, then also you will not see any signal. So, there are two possibilities I have said. Okay. Protein is very unstable, then you will not see much. Protein is super stable and requires high forces, then you will see it. Okay. There is another situation. So, if unfolding forces are smaller, are lower than the resolution limit of the instrument. So, in other words, in your force scale, okay, if you cannot resolve below 20 piconewtons, okay, so you do not see anything below 20 piconewtons. So, this absolute lowest is let us say 25 piconewtons. Okay. So, you cannot record, you will not get any spectra for a peak which is below 25 piconewtons. In that case, you will again see the same thing, you will not even notice. Okay. So, this is the other case where the unfolding forces are very low, then also your instrument cannot detect those forces. So, but still there is a broad area over which you can still operate and find out what is the unfolding spectra. Okay. Now, comes the aspect of designing a protein construct. Okay. So, I said that you will make this polyprotein. Okay. Now, let us say you have not gotten your signal, you know that there are 3 possibilities. So, these 3 are the possibilities why you may not be getting the signal. So, there is it is very difficult to find out which of these case applies to your protein. Okay. So, what is typically done to get around it, but you know that your protein is folding stably as per other experiments. Okay. So, what you do here is instead of just having a polyprotein like A, you make a polyprotein, okay, with A and B, 2 components, where B is a component whose unfolding signature has been previously established. Okay. The rationale being that if you are doing your experiment, then you know that because B has an unfolding signature, even if you do not get A, then you know that your B signal should always appear in your protein unfolding signature. So, in that case you can say that okay, probably A has very low unfolding forces, that is why we cannot detect it. Okay. Or so that is one possibility. Okay. Now, the next question comes is how do we design our multi-protein? Okay. Should we have a design like A followed by B, or AB, AB. Okay. So, in shorts, do I repeat A n times, B n times, or AB n times? Okay. So, n may be variable. So, you choose n such that at least you know that the protein is folding properly. Okay. So, your n might be 3 in one case, or it might be 4 in one case. Okay. But you need to make sure that this protein is folding properly. Okay. So, with that I stop here for today. So, we have seen what are the challenges associated with trying to ascertain the folding signature of proteins and assigning it to appropriate domains. Okay. Next class we will continue from here. Thank you.